Okay, I'm going to show you how to use single cell scatter SCE. So SCE is a so-called single cell experiment object. It's a new data type we created from a expression matrix and a gene list. For example, here I provide example data from merged S1, S2, SCE. So this data has been prepared before, and I will have another video to show you how to prepare for this SCE object. So after load this, you can see here, and there's SCE object. This is a single cell experiment object. It contains all the information you need. Now the next step, you will scatter plot. Remember, you had the scatter plot of SCE and take big X expression matrix and gene list as a input. But here you want to use another function called scScatter underscore SCE. So this function will take SCE variable, which is here, as input. And then you click return, then you will have this pop-up scatter plot visualization, and because all the information about the cell and their classification and many other variables has already been stored in the SCE object, so you have all the information here. So you can do the same as you have already done for the scatter plot for single cell, and you visualize this different clusters. And then you can, first button, you can see the expression of the genes. Select one and cross all the cell types. Okay, you still can rotate and look at where the gene is expressed. And then next button, you will be able to select to highlight the cell by their properties. So for example, right now the scatter plot is showing the cell cluster IDs. That's what you see here right now. But if you select another one, for example, cell cycle phase, and then the cells will be colored differently and based on their cell cycle labels. So blue ones are the cells in the G1 phase, and this orange one are in the G2 and or M phase, and this yellow or orange color indicated in the S phase. So you can rotate and see, okay, there's a cluster of cells in G2 and M phase, become separate clusters here. Okay, then depends on the properties you had. You have a batch ID. In this case, we have it merged two batch of data, so you can look at it, and you have a batch 1, batch 2. Batch 2 has more cells indicated in red. So that's the mixture of two batch of cells. So you can see this cluster is only contains the cell from batch 2 cells. Another button is to extract cells by clusters. Classifications. So if you want to select, say I want to select batch 2 cells, you see batch ID, and then it will ask whether you want to use batch 1 or batch 2 data. You see batch 2, and a new window will pop up. This contains only batch 2 set of data. If you want to extract, for example, cells in only G1 phase, you select this, and then this will be the cells only in the G1 phase. There are many other options you can explore and see its potential. And for example, this quick demonstration will be, you can switch three-dimensional plot with between three-dimensional or two-dimensional. So now you should be able to see the two-dimensional. Okay, data. You can change the marker labels and change the color schemas, refresh, and change it back to three-dimensional. Alright, I think that's give you a good point to start. Thank you.